for week 2 of the course Experimental Biotechnology. Follows. We will be looking at for each of the question. Take time to think, and whoever knows the answer can raise your hand. So we will work through all options. So um, we will work by eliminating the options in and that will help us know how each operands or operations are related. So the first question for today's session is the expression of electrophoretic mobility can be given as dash, and four different options are given. V is directly proportional to Z by R, V is directly proportional to E by R, V is directly proportional to Z by E, V is directly proportional to R by E. So whoever knows the answer can first uh, explain what electrophoretic mobility is, and then we can look at the different options. So you can think for a couple of minutes and then start raising your hands. Okay, here V is the velocity, Z is the charge, and R is the radius of the molecule. We can assume that if it's a spherical molecule, we can assume that it has a radius R. Hydrodynamic radius is different for non-spherical molecules. So when a molecule moves across an electric field, what primarily determines uh, the movement or the velocity is that how is the charges spread. So, and this can be approximately summed if we know the charge to size ratio, that is Z by R here. So if we have more number of charges for a molecule that is very small, then its velocity will be uh, higher. Like smaller molecules um, tend to move faster for the same amount of force. Here, the force is through uh, the electric current that is being passed. So, smaller the molecule for the same charge, still uh, the smaller molecule is the one that will move fast compared to the larger molecule. I hope that was clear. Agaro's gel electrophoresis is an example of boundary electrophoresis, horizontal gel electrophoresis. So you can take a couple of minutes to think about this question and then raise
So horizontal.
Can take a couple of minutes. We'll also discuss about other options first. So that was Akshita, right? What you can do is you can first start with eliminating the wrong options. That way we can discuss all the wrong options and then come back to the right option, so that we don't miss out on any of the options that are present here. So you can think for a couple of minutes and then start raising your hand, and then we'll discuss the answers. So you can raise your hand. So how this works is we can first, you need not type the answer in the chat window. Instead, what I would prefer is if we have a discussion, especially if we start with eliminating the wrong options here. So both Akshita and Rana, you can both of you each one of you can discuss one wrong option, and we'll eliminate those two and justify why those are wrong. So, do one of you want to unmute and tell what you think are the wrong options? So um, I would start with eliminating the wrong options that are here. For the first option that I would like to bring to your notice is option B, moving boundary electrophoresis. So yeah, Akshita, you were typing something. You can unmute. Yes. Okay, first we'll discuss what moving boundary electrophoresis is. In moving boundary electrophoresis, we don't have this physical constituent like a gel, either an agarose or an acrylamide. So we can instead you have these capillary or in some cases even in immunodiffusion assay you have these moving boundary electrophoresis method. So that is one of the options that is eliminated. Um, Vertical and horizontal are two different methods, two different orientations. So in vertical gel electrophoresis, we run them in the form of page. It could be either STS page, which is denaturing, or it could be uh, native page where you don't have any denaturing. Um, so page is uh, polyacrylamide gel electrophoresis. This belongs to vertical. In agarose gel electrophoresis, what we do is we run them horizontally for dissolution. So the right answer is horizontal, option D. And just one clarification, vertical is not used only for proteins. For RNA purification as well, people use polyacrylamide gels which are run vertically as opposed to the traditional horizontal method. So you need not assume that vertical is only for proteins. Even RNA and even DNA can run on, can be used for separating or resolving using page, not just proteins. The next question is dash is an initiator of acrylamide polymerization. Option A ammonium persulfate, option B temid, option C trisetzial, and option D acrylamide. So you can think about this question for a couple of minutes. Then, if anyone can tell me, if anyone can begin with the wrong option as we discussed, it will be easier so that we can learn about all the four different options that are present here. Yeah, Sukanya, Sai Sampath, Tanushri, and Akshita. Can any one of you unmute and then start with an incorrect option?
Citrus, it's the
Okay.
component of the enzymatic used for the catalytic polymerization.
Okay. What is the pH of tris that we use?
So it is evolving.
You can mention for both if you know. One of you mentioned that trisetial. So yeah, trisetial is um, actually trisetial is not your part. You instead you and STS as well. Trisetial is rather your buffer with which you make your gel. Nowhere related to um, your acrylamide polymerization. It just ensures the state of your glycine ion. pH 6.8 and pH 8.8, your glycine ion has different states. So that is what is happening with tris. And acrylamide is your actual component that gets polymerized and forms the polymer. So acrylamide is not the initiator. In all these cases of polymerization, we need something called as a free radical initiator. In this case, it's ammonium persulfate. It's extremely unstable. Monomer and acrylamide polymer. In between two acrylamide monomers, so interact to form cross-linking. That is how your pores are formed. Otherwise, these are just linear chains. Here, in this case, to answer this question, ammonium persulfate is the initiator of acrylamide polymerization. The next question is the full form of TEMED is dash. And you have four options. Option A, N tetramethylene diamine. Option B, N prime tetramethylene diamine. Option C, N prime N prime tetramethylene diamine. Option D, N prime tetramethylene diamine. You can think about this question for a couple of minutes, and then this is straightforward. So, um, if anyone wants to answer, they can unmute. Does anyone want to answer this? Full form of TEMED? Right, it's option C. This is the structure. So we have two nitrogens here, because of which, and two nitrogens, and each of the nitrogen has two methyl groups. That is why one of this is denoted as N, the other one is denoted as N prime. Right. The next question is the pore size in polyacrylamide gel is determined by option A, ratio of the concentration of acrylamide and bisacrylamide. Option B, amount of APS used for gel formation. Option C, amount of TEMED used for catalysis of the cross-linking reaction. Option D, ratio of concentration of APS and TEMED. Okay, so in order to explain this question, you need to first tell us the components and what each component component's particular role is. We have earlier discussed that. You can take a couple of minutes to think about this question and then begin with the answer. Please do not type the answers in the chat box. You can think about this question for a couple of minutes and then we'll okay, Tara, I hope you had answered for this question, the precise question. Do you want to explain why you have typed option B? Tara Panwar? Yeah, you can unmute and then you can tell us why you think it's option B. Or if anyone else want to answer, they can also tell us why a particular option is incorrect. Okay, you had mentioned for the previous question. Okay, it's actually uh, not B, it's option C for the previous question, N prime, N prime. This is your right answer. Okay, do you want to explain why you think this is the right answer? Before you go to this, you can mention what acrylamide is, what bisacrylamide is. Although we have discussed this in the previous set of questions, it would be better if I get to know whether you guys understand this. Does anyone want to unmute and answer? Okay, I'll start with eliminating the incorrect options here. So the first option that we would eliminate is this option D, ratio of concentration of APS and TEMED. Here we know this is the free radical generator, and this helps in that process. So by changing the ratio of APS and TEMED, we would just either fasten the reaction or slower the reaction. If we have more of APS and TEMED, we'll have a faster reaction, faster polymerization. If we have less of this, we'll have slower polymerization. So that has nothing to do with the process. Then option C, amount of TEMED used for the catalysis of cross-linking reaction. Again, no. Amount of APS, again, no. So the answer is acrylamide, bisacrylamide. So one thing that you need to remember is these acrylamide monomers can polymerize to give these linear chains. whereas this bisacrylamide can stand between two linear chains and it can form this cross links. So assume this is this way and you have another green here. So that would mean this is the only pore. So depending on the number of or the ratio of bisacrylamide, then a pore size can be decided. We use a ratio of 29 is to 1 or 30 is to 1. So the next question is, if the amount of cross-linking reaction is more for polyacrylamide gel, the pore size will be dash. Smaller, larger, unchanged. And option D, no change in pore size, but the number of pores will decrease. And then you can think about how cross-linking would affect the pore size and then justify why one of these options is right. Okay, so like we discussed, there uh, have one more crosslink after even the pore size will be small because we have more of cross-linking, the pore size will be further reduced. Uh, More for polyacrylamide gel, the pore size will become smaller. And the pore size decreases as the number of the pore size decreases as the number of pores increases. Here, if there is only one crosslink here and only after 10 such repetitive units, if we have another crosslink, there will be only one large pore. But if we have this crosslink after every two repetitive units, you will have more than five pores here, and each pore will be small. So it's actually one large pore getting constricted into multiple different small pores. Of bromophenol blue, A, stain the proteins, option B, stack whoever wants to answer. Sorry, who's this? Okay, precise. Okay, okay, what is staining a protein would mean? You can unmute. We can discuss this.
come up
Sorry, who was that? And what did you mention? It was not clear. Who was that? Akshita. Okay, Akshita. What did you want to mention? You can unmute and talk because that way it's easier to know what you're thinking rather than typing in the chat window. Okay, mic is not working. Okay, what about Sai Sampath? Okay, I'll. Hmm. Okay, first we'll eliminate the incorrect options. Uh, so checking the pH of the sample. So this is not a pH indicator. We are not checking the pH of the sample, and we don't care about the pH of the sample here because that would in no way affect what is happening within the gel. We have two defined pH within the gel. One is 6.8, the other is 8.8, and that is what would define how the migration happens. So we don't have to worry about the pH of the sample. That is um, so option C can be eliminated, and option B to stack the proteins properly. So for this purpose only, we have a stacking gel followed by a resolving gel. So it has nothing to do with the loading die per se. And option A to stain the proteins. So if we want to stain our proteins, then if we are going to allow our protein to run throughout the um, gel, then we would ideally see a gel with full of blue colors. But rather, that is not how we see. We stain the protein using some other thing called as coma C brilliant blue. So this is the staining solution that is used, and once it is stained, it looks something like this. On the top here is my stacking gel. The bottom here is my resolving gel. These are different fractions. This is a protein marker of different molecular weights, and this is my protein of interest. So whatever was in the sample was a mixture of all of these, and I see that my protein is running at around 45 kilodalton here. Right. So the actual goal of this bromophenol blue is to see whether my gel has my proteins have migrated towards the end of the gel. So when we run the gel, we don't see this. It's a transparent gel. We want to know if whatever sample we've loaded here have come towards the bottom end. Because only if it has come towards the bottom end, we will be able to resolve all the proteins of different molecular weights. To confirm this, we use bromophenol blue. So option D, to understand the progression of proteins through the gel, is the right answer. Next question is, during stacking, the sample gets sandwiched between, and four different options are given here. You can think about this question for a couple of minutes, and then we'll discuss how this stacking happens. Okay, Akshita. So you had mentioned about glycine. Is there some other element that we are missing here? Okay, there are two things that you have mentioned. As in stacking, the pH is low, so glycine moves slow. But as it enters the separating gel, both acquire same charge. Stacking is important as it helps the sample to enter through the same point. Right, we'll go through whatever you have told. So, what all do we have in our running buffer is we have tris chloride, tris HCl, which has chloride ions. We also have glycine. These are the charged entities. So your molecule is this way. You have your protein that is linear because it is denatured. You have chloride ions and you have glycine. Now, like it was mentioned, at pH 6.8, glycine stays back, and it your chloride ion pushes glycine and pushes your protein to between glycine, and glycine is slow, that is rate limiting, whereas in your resolving gel glycine is fast. This page, the resolution is based on the dash of before you go towards answering this what each component that is involved in STS page does. That will help us answer this question. Okay, what do you mean by size, Tara and Akshita? I'll discuss the different components on STS page. The word STS means sodium duodecyl sulfate. As you have seen here, what happens is for every two amino acids, this sodium duodecyl sulfate goes and binds specifically, and because of this, your protein is evenly charged across its length, and it is denatured, which means it doesn't have its 3D shape anymore. It it virtually functions as a 2D linear stretch of amino acid which has SDS attached to every two amino acids. So that is what happens in SDS. So now, because the length of the entire protein and the charge across its length is constant, for different lengths of protein or different amino acids, different um, yeah, different molecular weights, we have a corresponding charge. So, like we know, this depends on. Uh, so now the only factor that determines if the protein can move is size, like it was mentioned. But the size again depends on the molecular weight specifically. There is no shape associated with it. There is no electrical charge associated with it. Both of it is now normalized. So that is what I had asked us what you mean by size here. So molecular weight represents the size of the protein. So it's rather molecular weight and not the size. I hope this was clear. Yes, page experiment is performed. Covers 4.5 and you can take a couple of minutes to think about the question and then mention what RF is and how to calculate RF. Akshita has mentioned, she has mentioned that it is the distance traveled by the protein upon solvent. Uh, okay, it's not the solvent, it's the solvent friend. And in STS page, we talk about difference. Um, so can anyone recall what the different is? We had an earlier question on the same part. Here, we mentioned about bromophenol blue. So we allow the bromophenol blue to come to the end of the chain. So usually when I load the protein samples here, I allow the protein samples to reach the end of the chain. So that is where my different is. And my protein has, so the distance traveled by the different is calculated from the start of the resolving gel to this. Whereas the distance traveled by my protein is from this to this. So this is your RF. It's also called as retention factor. Usually it's not used with the STS page, but rather for thin layer chromatography or paper chromatography methods, where you dip, you add your components that has to migrate and you dip it in your mobile phase and they separate and you calculate what the retention factor is. It is not usually used for STS page, but the way you calculate is similar. You allow your solvent friend, use that as your standard. In this case, it's 10 centimeter, and compare to your solvent friend, how much is your protein cover? So 4.5 by 10, it's just 0.45. That is how Sukanya Maiti has mentioned 0.45. Good. The next question is, de-staining solution is used for dash. Removal of CBB staining solution from protein lines. Removal of CBB staining solution from bound non-specifically to polyacrylamide gel. Removal of bromophenol blue stain. Removal of stains due to contaminations. You can think about this question for a couple of minutes and then answer. And also before you mention what the right answer is, if you can give the component of de-staining solution, how is de-staining solution made, that will make more sense because I will be discussing how the de-staining solution is made. Okay, so first your de-staining solution is made by adding Kumasi brilliant blue, methanol, water and acetic acid. And your de-staining solution doesn't have Kumasi brilliant blue, it only has methanol, water and acetic acid. Uh, Once your gel finishes running, you would put the gel in a solution containing pumice brilliant blue, methanol, water, and acetic acid. So what this does is it stains multiple regions, like what was mentioned. If you see here, this is only the gel, and what was transparent earlier is blue now. But what we don't want this. We don't want the entire gel to look this way. We only want the proteins to take up. So we remove the non-specifically bound pumice brilliant blue by adding the de-staining solution. So option B is the right answer, and this is what Akshita has mentioned here. Good. The next question is ETBR is used for the staining of dash DNA, protein, RNA, and lipids. So here, you can think about this question for a couple of minutes. You can expand what ETBR is and what are all the possible macromolecule biomolecules that can bind with ETBR and how it binds. So you can think about this question for a couple of minutes and then answer. So okay. So what ETBR is? ETBR is full form of ethylene bromide, like Akshita has mentioned. So the structure of ETBR looks like this. It has these triphenyl ring attached to one. Now, how does this interact? So it interacts with DNA bases in the form of intercalation. So we all know how benzene rings are present. So similar to the phenyl rings here, if you have a stacking interaction between two bases, so okay, so in both DNA and RNA, we have these bases that are stacked. Now, this ETBR goes in between these two stacked bases. This process is called as intercalation, and it has an improved fluorescing property when it is intercalated than when it is natively present. And because of this reason, it can bind to both DNA and RNA. RNA, although it doesn't have double strand like DNA, it can form secondary structures and tertiary structures, which involves the formation of hairpin loops and um, other, I mean, basically the formation of hydrogen bonds here. So that is why ETBR can bind with both DNA and RNA. So it is not restricted to only DNA. And this major groove and minor groove is not specifically relevant here because major groove and minor groove is a large space and your ETVR is not that large. ETVR rather intercalates, so that is how it binds. Major groove and minor groove is for binding of proteins that bind to DNA. For example, transcription factors like repressors and enhancers. That is what binds to the major groove and minor groove. Was that clear? Okay. The next question is, in native page, the sample gives a molecular weight band of 189 kilodalton. In STS page, the sample gives two bands of molecular weights 55 kilodalton and 67 kilodalton. The protein is dash homodimeric, heterodimeric, heterodimeric, heterotetrameric. Think about this question and the differences between native page and STS page. So to start with, you can tell what happens in native page that is different in STS page, and then we'll decide how to go about with this question. So in a native page, we don't denature the sample, which means multiple different chains, even if they are not covalently linked, they will ensure that your structure is maintained. But in an STS page, this is not happening. And they say that there are two bands, one at 55 kilodalton and one at 67 kilodalton. And but in a native page, it gives 189 kilodalton. So you need to find out how many coefficients of 55 kilodalton plus how many coefficients of 67 kilodalton gives you 189 kilodalton. So um, obviously 55 plus 67 is not 189. The next way to see this is 52 into 55 plus 1 into 67. 2 into 55 is 110 plus 67 is 177. That is not the case. So 1 into 55 plus 2 into 67. So 2 into 67 is 134. 134 plus 55 is 189. So this tells us that the 67 kilodalton protein acts as a dimer and 55 kilodalton is a monomer. And because there are two different chains, it is a heterotrimer. Okay, there are three chains, two chains of 67 kilodalton and one chain of 55 kilodalton. And because these are different, it's a hetero. And because there are three components, it's a trimer. So answer is option B. The next question is, agarose acrylamide composition gels are used to analyze protein samples of molecular weight dash. Option A 20 to 100 kilodalton, option B 100 to 200 kilodalton, option C 20 to 500 kilodalton, option D 500 to 2000 kilodalton. So you can take a couple of minutes to think about this question. So to think about this question, we usually run acrylamide gels for up to 500 kilodalton, whereas agarose gels is where we can run from in base pairs, and these are extremely large, above 500 kilodalton easily. So how you need to calculate this is, one base pair is 660 daltons. So if it was 1 kilo base pair, 660 kilodalton. 1 kilo base pair is 1000 base pair, and that is very small. Plasmids that we run have 4 to 5 uh, 5 kb kilo base pair, 4000 to 5000 base pairs. That is easily more than 2000 kilodalton. So when there's a composite gel containing both agarose and acrylamide, it can be used to run 500 to 2000 kilodalton. Shown here as ferritin. So macromolecules. Such molecules which your polymerization happens reduces. Solely because of this, when we are casting our gel, we pour our resolving gel, add a layer of isopropanol, and ensure that no oxygen from the atmosphere enters the gel. So this is how it is done. And the answer for this question is polymerization reaction. Right. So with this we come to an end of today's set of questions for week 2. If anyone has any